This video is on cell development. How does a haploid egg cell with 23 chromosomes and a haploid sperm cell with 23 chromosomes that unites in fertilization to become the zygote, this fertilized egg with 46 chromosomes, how does this zygote become a fully developed organism with specialized tissues? Well, the first thing that happens is this cell needs to divide. So we undergo mitosis, and at first we get to this 8-cell stage. This 8-cell stage is made up of specific stem cells known as totipotent stem cells. Totipotent stem cells are cells that can become any type of cell given in the body given the proper signal. So as I said before, these totipotent stem cells can become any cell type if given the proper signal, and proper signals can include transcription factors or growth factors. So at this 8-cell stage, these cells can become any type of cell in your body. These cells continue to divide by mitosis. As they divide by mitosis, we get the blastula, which is basically a hollow ball. The cells are on the outside and the inside is hollow. These types of cells are also stem cells, but they're on their way to specialization. Okay? So they are called pluripotent stem cells. Pluripotent stem cells can become many cell types. For example, maybe they can become any type of cell in your intestinal tract. Okay? So they can't become every cell type, but they can become many cell types. Both totipotent and pluripotent stem cells are examples of embryonic stem cells found in your embryo. As you continue to develop, these cells specialize even more. So as you get older, you can also have adult stem cells. Adult stem cells are multipotent, meaning they are specialized a little bit to the type of cell. So they can't become any type of cell, but they can become several different types of cells. For example, a hemopoietic stem cell or a blood stem cell could become a red blood cell or a specific type of white blood cell. So now the question is, how does this zygote that's dividing and causing this organism to grow, how does it ultimately become a specialized organism? So how do stem cells become specialized? This occurs early in development, and it occurs through control of gene expression. We turn genes on or off to make specific proteins in specific cell types. For example, a red blood cell might make a hemoglobin protein, right? Where a skin cell might make a collagen protein. So different cells make different proteins, even though they use the same DNA, in order to specialize. We're going to talk about how this happens. This can happen through internal and external signals. So go ahead and flip your big sheet over, and we're going to start by talking about cytoplasmic determinants. This is an internal signal, and I want to talk about what they are, where this happens, when this happens, and how this happens. And how does this lead to cell specialization? So right here you see a sperm cell and you see an unfertilized egg cell. If you look closely, you'll notice that there are chemicals inside this unfertilized egg cell. And I'm color coding them just so you can see. I'm going to zoom in a little bit so you can see them. And if you notice, these chemicals are not evenly distributed throughout the cell. The left side of the cell has more of these pink circles. The right side of the cell has these yellow triangles. These chemicals are known as cytoplasmic determinants, and they are not evenly distributed throughout the cytoplasm of this egg cell. So because the cytoplasm of this egg cell is not homogeneous, there are varying amounts and locations of mRNA, transcription factors, and enzymes. Once fertilization occurs here, and we have our zygote, we have our fertilized egg, there is still an uneven distribution of these cytoplasmic determinants. So that when this cell divides, when we have cell division here, here are two cells. If you notice, the chemicals in their cytoplasm is not the same. And if these are transcription factors, these can activate different genes, leading us to move towards specialization. Some of these transcription factors could activate genes and be specific activators. Some of these transcription factors could silence genes and be known as silencers. So after division, the new cells have different cytoplasmic materials, okay? And this different chemical makeup in the cell's cytoplasm can lead to cell specialization. That's why they're known as cytoplasmic determinants, because based on what you have in your cytoplasm of the cell, that can determine what cell type you become. And one important note to remember is that these activators and silencers in the cytoplasm are an internal signal. These internal transcription factors are going to activate or deactivate certain genes to increase or decrease protein synthesis of those genes. These transcription factors will activate or deactivate specific genes to increase or decrease protein synthesis of those genes, right? So let's talk about an example of cytoplasmic determinants. This is a very famous example. It's all about the bicoid gene in fruit flies. Okay, so right here you see an unfertilized fruit fly egg. Okay, and the bicoid gene right here, this is the bicoid mRNA, is this darker region. After fertilization, now we see an early embryo here, and once again, when we look at this embryo, we can see the production of that same bicoid protein at the same side of this embryo. Now, when they look at the mature fruit fly, what they notice is that where there's bicoid, genes are turned on to produce the head of the organism. Where there's no bicoid, the cell automatically produces the tail or the abdomen of the fruit fly. So scientists did an experiment. What they did, their claim was that presence of bicoid allows for the development of the head. But they needed evidence and they needed reasoning. So they altered some fruit fly larvae. In one experiment, they took out all the bicoid. No bicoid at all. And what they found out is that fruit fly produced 2 tails and no heads. In another experiment, they added bicoid to both ends. And those fruit flies ended up having 2 heads and no tail. So their claim was that the presence of bicoid leads to head production in the fruit fly. Their evidence was that wherever there's bicoid, there's heads. Where there's no bicoid, there's no heads. And the reasoning is that this bicoid protein is probably a transcription factor that turns on the genes necessary to produce the head structure of the fruit fly. And in the absence of bicoid, it naturally produces the tail of the fruit fly. Okay, so this is a very famous example of cytoplasmic determinants in fruit flies. Let's look at an external signal, which would be induction. I flipped back over for a second because you're most likely to see cytoplasmic determinants having huge impacts at this 8-cell stage, because basically the cytoplasm of the cell is going to help it become differentiated and decide what type of cell to move into. When you get to this blastula stage, the cells start to talk to each other. So that's our second example of how the cells can become differentiated, can become specialized. And that example is due to external stimulation from other cells, and it's called induction. Okay? So if this is cell 1 right here, and this is cell 2 down here, cells can send messages to each other. Okay? Cell 2 can send a chemical message to cell 1. Cell 1 can send a chemical message to cell 2. And that can help with differentiation. So basically, these cells can release signals that travel to other cells, and the cells can talk to each other, which can start a cell signaling pathway that leads to a response. And the response we're really looking at here is differentiation and specialization. So it could be a nuclear response turning on and off genes to allow the cell to specialize. Right? And you can see the blastula right here. Sometimes the message can even be something like a growth factor, telling a certain type of cell to divide more often. A really classic example of induction is in C. elegans. C. elegans is a worm. They studied C. elegans very closely, and they looked at this 4-cell stage. So here's cell 1 right here, here's cell 2 right here, here's cell 3 down here, and here's cell 4 over here. And these cells actually talk to each other. Okay. This picture right here shows these 2 cells talking to each other right here. Okay? And basically, cell 3 right here, cell 3 is getting messages from cell 1 and cell 2 on this side. So this side of the cell, when the cell divides, this cell right here will have mainly received messages from cell 2 and cell 1. This side of the cell has mainly received messages from cell 4 and cell 2. Right? So this side of the cell received messages from cell 1 and 2. This side of cell 3 received messages from cells 2 and 4. What happens because of that? Well, when this cell divides, this cell in the division will go on to form muscle and gonads, whereas this cell in the division will go on to form the adult intestine. So just by being on a specific side, by having cytoplasm from a specific side of the cell when the cell divides, will lead these 2 cells to become specialized because of what cells they were near before division occurred. So these Hox genes are also known as homeotic genes. These are master regulatory genes. They regulate gene expression, leading to cell specialization. Different Hox genes are active in different cells, which determines your body plan. For example, the plan for where your head, your appendages, your abdomen, your tail is. So for example, this Hox gene right here is active in this part of the embryo, which will code for the head. This Hox gene is active in this part of the embryo. These Hox genes are activated in the thorax of the embryo, and they help make sure your body plan is in order. So your head, your thorax, and your abdomen are where they're supposed to be. Hox genes are highly conserved. This means they are similar or the same across the animal kingdom, right? Like the Hox gene for vertebrae is going to be similar in a snake and a rabbit and a human and a gorilla. They're all going to have similar Hox genes, okay? This example is a fruit fly chromosome. These are the Hox genes on a fruit fly chromosome, and this is the area they correspond to and are active in a fruit fly embryo. So I said that homeotic or Hox genes are examples of master regulatory genes. But what is a master regulatory gene? A master regulatory gene is a gene that produces a protein that turns on or off other genes, which can lead to specialization, meaning the cell becomes a neuron or a red blood cell or something like this. Cell specialization actually has a couple different steps. So I'm actually going to put a line here, and above this line we have our unspecialized stem cell. Okay, here's our embryonic precursor cell. Here's the nucleus. You can see in this stem cell, right, this master regulatory gene is off in the DNA. Other muscle-specific protein genes are off. So this is just a stem cell. But how do we get this stem cell to become specialized, right? Our end goal, let's say, is a specialized muscle cell. How do we get there? Well, there's two steps. The first step is known as determination. And the second step is known as differentiation. Determination means it will become a specific cell type. So for example, if this stem cell right here gets a signal, and that signal causes the mRNA for this master regulatory gene, we've turned the DNA on, which has allowed us to transcribe the mRNA, and it's allowed us to make this MyoD protein. This is right here, this protein right here, is a master regulatory protein. So once we've turned this gene on to produce the mRNA, to produce the transcription factor protein myosin D, we've determined that this cell will become a muscle cell. It's like if you have a big chain of dominoes lined up and you knock over the first domino. Well, all the dominoes haven't fallen over yet, but you've started the process. So in determination, we've started the process. So that's this step right here. Okay? But what is differentiation? In differentiation, it has become a specialized cell. How does that occur? Well, here's our DNA here, right? This is our DNA here. And this myosin D gene has already been turned on, right? Our regulatory protein is already on. So the DNA is on, it produces its mRNA, and the regulatory protein has been produced. Well, this myosin D regulatory protein is going to go and turn the DNA on so it can produce mRNA, so it can produce another transcription factor. This transcription factor is going to go to other locations on the DNA, turn this DNA on so we can produce the mRNA, and we can produce other muscle proteins. We can produce myosin, and we can produce actin, and we can make our specialized muscle cell. So that's the process of how cell differentiation and cell specialization occurs. Thanks for watching the video.